Mhm. Okay, so let's start. Today we're going to talk about mainly hypothesis testings. It's not really unrelated to whatever we were talking so far. We're going to just explore a little bit more on this. I'm going to wrap it up more faster than other classes because I want to keep it short, because what I would like to do is to give a little bit time to you to talk about what you did with that COVID-19 homework. So we go from there, okay? So and if I mentioned you have to register to the, basically you have to register in the Zoom with your email link, email address, official email address, so you don't go to the waiting room, because I'm worried if you go to the waiting room, I don't really notice that. Okay, I try to, but it's better to register so you don't stop in waiting room. So, okay, before we start, let's, I have some announcement. I'm going to, as you know, the project 2 is available. Let me show you. So as I mentioned in the Canvas website, in the Canvas for the course, you can see all the assignments. Okay. If you go to the homework, I mean the homepage, you can see all the necessary links here. Recorded session, assignments, the solutions that I'm keeping basically uploading here. You have some resources here. You can check. I'm going to upload the sources here. So if you go to the assignment, you can see all the assignments that we are working with their deadline. So lab 6 is going to, basically we are going to work it today, and then I'm going to give you until Sunday midnight to finish it if we couldn't finish it today. The homework 5 is already in the system. You have time until April 14, which is next week. And the important one is basically project 2. It's already available there. I uploaded last week. I forgot to tell you about it. So it's available. The checkpoint is the next week. Okay. So you remember we have like a checkpoint in the middle of the project and then the final submission. So you can check all the deadlines here. Okay. Clear?
Yes.
Yes, teacher.
So
Yes, teacher, good.
any questions about activities? No? Okay, so just make sure you keep checking this. I'm announcing it during the class, but you make sure you also check this and you're basically aware of what's happening, okay? I had some emails from students that they had problem accessing Citrix, accessing basically the server through the Citrix. I have also sometimes problem. I have to like click, wait. Sometimes it doesn't work. I have to try again. Sometimes it works. Sometimes it doesn't work. It's a little bit problematic. That's why I really recommend you to do it on your local machine. It's really easy to install it. I did it. You can run it everything smoothly on your system without any problem. So try that. It would make your life much easier. So about these are the submission I received for COVID-19 homework. So I think they're all submitted, or maybe there are some missing here. I don't know. I'm going to double check. We're going to talk about it. So let's continue. Let's see that. Okay. You remember last class we were talking about the case of Swain in Alabama, the court case, that we were trying to see if the jury panel were selected randomly. It was basically a fair panel for this the guy Swain that we mentioned, he was accused of raping a white woman. So he was a black guy. So, and we talked that most of the jury were white. Basically, all of them, they were white. And I mean, the initial panel were basically 8 black. Then they had to basically leave. Anyway, we wanted to basically study if that 8 is a fair, basically, selection from the society that we had. So we talk about it. Basically, that's a rule. Basically, the jury service shall be selected at random. So that was we were trying to basically study. We were trying to study the randomness of that selections. So let's just review it very quickly. Again, I'm mentioning, as you can see, I'm running it in my local host. Try to do it. It's much easier. It's much faster. It makes everything much easier. And another thing is Google Colab. If you heard about it, you can also run your notebooks on there. It's not that, for example, when you're trying to load some files, then you have to have the files in your server. I don't think it's going to fetch from Google Drive, but you can try that also. It works there too, but not just OKPy commands or checking your codes doesn't work. So basically what we were trying to do here was, that was basically the panel that they selected. There were 8 out of 100 were black. And we were trying to do basically, we started with distribution of eligible jurors and the actual panel. I'm just going to continue. I'm not going to really talk about the codes. They're available, not really hard to understand. So basically, this was the table. This was the eligible people in the. Okay.
Jury?
No, wait a minute. Let me see if I can find. Anyway, these are the. This was the available. Basically, these are the population, the distribution of the population. We had 26. 26% of people as like black and 74% as other races. But the panel was consisted of 8% black and 92% basically white. So we define it, okay? Then we define a function to basically draw panels at random from the population that we had. So we get, this is for example, one run of this. If I run it again, you can see the random is changing. So basically what we are doing, we are picking some random 100 people randomly from our system, basically samples. And then we counting how many of them are black and how many of them are from the other races. So all the whole idea was to study if the jury selection was fair. Then we talk about total variation distance. Basically, the point about it was how different is the distribution of basically our samples and the distribution that we get from our, for example, in this case, the panel, okay? So we got these numbers from comparing this eligible with the panel. Basically, that was the whole code of it. I mean, don't get confused with the codes here. So basically, we divided them. We got this, we minus this, and we get the sum of the both basically raw, and then we divide it by 2 to get the TVDs, total variation distance. Then we define a function, so because we're going to use it later. Then what we did, we run the experiment like 1,000 times, and we get all the basically all the TVDs, and then we did the histogram. So we got this, and that was basically this is the TVDs from this jury that was selected in real life, and this was our experiment of picking the juries like 1,000 times from our society. So then we discussed that comparing these TVDs that I get here from basically the real one and comparing them with my experiment, then we saw like, for example, mostly the TVDs are falling here, 0.1 above, 0 below, okay? But that real one was here, like 0.18 or basically 18%. It is like somewhere around here. So you can see how unlikely was that selection from the society, from basically how unlikely was getting that jury panel for that case. That was what we discussed last week. So let's study another case. It's more or less the same case. It's the same basically scenario. That was the Swain case was happened in 1960. So you may, I mean, if you know the basically background, the history of United States, you may say, okay, that time it was normal not to be fair due to the racism that existed that time. But what about now? What about what's happening nowadays in the recent years? So American Civil Liberties Union, or ACLU, released a report on racial and ethnic disparities in Alameda County. Alameda County is a county in California. So they interviewed a panel of 11 different felony trials from 2009 to 2010. So in this, basically, this duration from 2009 to 2010, they studied 11 different felony trials, and this is the basically data they got from that study. So basically, these are the distribution of the society in that county, Alameda County. So basically 15% of them are Asian, 18% Black, 12% Latinos, 54 white, and like 1% as other races. So they studied this panel of like 11 different felony, and in total they studied 1,453, this is the size, of the people from the panel, and then they see their distribution about the races. So they see the panels are like 26% Asians, 8% Blacks, 8% Latinos, 5% whites, and 4% like other races. So again, looking at this, you might feel the same situation is happening, the same situation that we had with the Swain case in Alabama. But we cannot just look at this data, look at the table, and just conclude just like that. Okay, we need to basically analyze and maybe going through the same process to see if it was a fair distribution or not. But this is the basically case. This is the bar chart of this data, this table. So you can see, like for Asian, we had much more in the panel than in our basically county. I mean, considering the percentage. So it was much lower for the Black, much lower for Latinos, and for white people it was equal, and like others it was different. So let's study this case with this, doing the exactly same method, and see what we can conclude from this particular example. So again, what we do here, from the panels table that we have defined already, this table, we select the ethnicity. From the ethnicity, we select basically 10,000 samples, and we group them by ethnicity. So you can see if I select this type of samples, there are random samples from my table, from my basically ethnicity, and if I group them by ethnicity, you can see the distribution here. So it's almost like a fair distribution between all of them. But you remember I mentioned this is not the case because the ethnicities, they don't have the same weight. They have different probability of picking. So this is not the right way to pick from that. So this was the eligibility of basically in our county about the people, how many percentage of each group we had. Then the same thing that we discussed last time, that you need to put the weight, okay? Basically, we are defining this weight as the eligibility, basically percentage that you saw in the table, the panel table. So so here again, we do the sampling from this table, but now we introduce the weight, we group them by ethnicity. So now you can see the distribution is more or less like what we expected from our basically table, okay? This, as you can see, you can buy like 54, others are much lower, like Asian 15%. So if you compare this, you can see we are getting more or less the same things. For example, for other, we're getting around the 1% of the distribution. But there is an actually easier version, simpler way to do the same things using this proportion from distribution. If I just run this, get the help, you can see this is the help documents for that function. You can go through it. You can see you're introducing a table, label, sample size. Then we have this column name that is samples. So you can see how it works, but it's not complicated. It's just like this. We have our table, we have the weight here, and we have the size of our basically panels that we are selecting from the table.
Teacher.
Yes.
So the weight is taken from the same table?
Yes, from the panel table. The panel table, if was this, if I put it here. This is the panel table, okay?
But I mean with the function proportions from distribution, you just like add the label of the column.
Yes, it just worked like this. I mean, basically what we do here, you remember here, we pick the randoms, then we put the weight, we define the weight, then we get another basically, like let's say table, then we had to append it to our original table. We had to go through all this basically process. Where was that? What we did, for example, in this case, okay? But this is just a simpler version of doing the same thing, putting the weight, selecting basically a random from your table and like appending it to your table.
Okay.
Okay, so it does basically all those things at the same time. It's much easier to use these functions. Is it clear?
Yes.
Okay, so if we do this, so we get this distribution. So you see here, not in a percent format, so we can basically put this table in a percent format. Okay, it's here. So we can basically change the basically represent the data in a percent format using these commands, if you remember, set the set format. So now you can see if I do the same this distribution now, this was the basically table. We have this races, we have this eligible, basically the population, how distributed is the population. These are from our study basically. These are what happened in that 11 trials, Baloney trials that we studied, like 1,400 something cases, and this was the distribution in real life. This is what I'm doing, basically sampling from this part, from our population, and then I show what's my distribution. Is it clear so far? Okay, so if we do the bar chart from this table, what we see is basically this type of distribution. These are basically the eligibility, the population of our basically our populations. These are the panels. The yellow one is the panel that are selected from basically in that 11 trials. And this yellow one, the like light yellow one, is my random samples from the random samples that I took from this basically my study case here. So what do you think? Just looking at this bar chart, can you drive some conclusion from it?
The eligibility have to be equals to all the population.
I mean, eligible is my distribution in the population. Okay.
Okay.
So I know I have this county. There are like 15% of the population, and the county is like Asian, 12 are Latinos, like 18% are Black. So these are my real basically county.
Okay, that comes from the table, and that it's calculated, but in the real life, it have to be, the eligibility have to be equals to anyone, no?
So no, eligible is like, you remember here, if, let me show you this one again. You remember these are the basically process of picking a jury from our trials. So these are eligible jurors in a county, okay? Anyway, we have a city, okay, that there are different races living in the city, and they are not distributed like exactly like you have 20% Latinos, 20% Black, 20% white. So they are different numbers. So these are the eligible jurors. So I have like 15% of my eligible jurors as Asians, 18% as Black, and so on. Okay?
Okay, yeah.
It's clear?
Yes.
Any question? So what do you think, guys? Just looking at this, can you drive some conclusion from looking at this bar chart, or we need some more data, or we can roughly say something about it? Any comments?
I think not.
So you need more information. Basically, we can see that the panel is a little bit different with what we selected randomly from the society, but we just did it one time, okay? Here we just run it one time. This is one sample from my society, but that one sample is more or less the same distribution with the society, but we cannot just, maybe what I can say is we cannot just judge based on this particular sample, so I need more data. Let's go back to the data. By the way, this is my cat. His name is Wheeler. Okay, so now you know my cat. So okay, we talked about like two type of like different type of distribution last time. We talked about like probability distribution, empirical distribution. So today let's talk about distribution of a statistic. So let's define this type of like terminology so we don't get confused. So like a statistic can be defined like a quantity computed from particular samples. Distribution can be defined like the chances of each outcome of the sampling, of the sample that we get here. And then sampling distribution can be chance of each value of a statistic computed from all possible samples. So that's our sampling distribution. So this is also known as a probability distribution of the statistic. Okay. And empirical distribution is the observation of the statistic. It's computed from some samples drawn at random. So in that example, for example, that was our empirical distribution. We had basically, we computed some basically samples we drawn drawn randomly from our basically samples. And then we studied that, we observed those study, that's our empirical distribution. If we talk about same thing in, for example, this Alameda County case, our statistic is what we want to study basically that TVD between a sample and the population distribution, because that's how we could say, was it a fair basically selection of the juries, or it was not fair, it was not total random, there was other basically things was happening there. The distribution is the chance of each possible TVDs. That's our distribution. And the sampling distribution is exact probability of each TVDs in our experiment. And then empirical distribution, talking about this example, is the probability of each TVDs using simulated panels from the population. So we define these things, and now we're looking at this basically how we basically defining this terminology, let's say, in this case. Okay, so we are going to look at the TVDs, and because that's how we were deciding about the fairness of the distribution or not. So it's the same thing that we did, defining basically the function, the total variation distance, TVDs, and calculating this. This is basically the TVD between my eligibles and the panels, the actual panels that I observed. Okay, not not my samples, the actual things that was happening. This is the TVD between the eligible and my random samples. Okay, if I put the table here, you can see this is the table. So basically, this is between eligible and the panels. Okay, this is the TVD. This TVD here, this one that you see here, is between the eligible and the random sample that I took from my basically county. This is the same thing. Sorry. So this was just we did here one time, one selection of 1,453. But what we want to do, we want to basically experiment that with multiple try, not just one try. We want to try it multiple times and see what information we get. So if I do the same process like 1,000 times, okay, and I basically pick that 1,453 person from my basic county and then calculate their TVDs. So I put it in this table. The name is results. So if I now do the histogram, you can see this is the distribution of this case. Okay. So now what you can say from this. So remember now we did the same thing 1,000 times, okay? And these are the results that we get. These are the distribution of TVDs. This TVD is 0.14. Basically means somewhere around here, okay? The TVDs of our actual panel. So now what you can say from this? Was it a fair selection or unfair selection? Anyone, comments? Just say what you think. I mean, no worries, it's not right, it's wrong. I mean, there's no wrong answer here.
I mean, comparing to the other case where the jury was only composed of white and black people, the ratios do make more sense at a first glance in this case. But if we judge based on this table of the PVDs, it still makes no sense that was the result.
So I didn't get. So you mean it was not a random selection of the panels? It couldn't be random.
This table, it doesn't appear to be.
Okay, true, because what we see, what we observe here is like the TVD that we get is basically this is the distribution. So mostly they're happening around 0.025. Basically, as close as they are to the zero, it means they are how to say, they are more fair. Okay, TVD should be closer to zero here. It means our samples is representing exactly our how to say, our count in this example. But the one that we saw here, this is the number that they got. The panel is like 0.14, which means around here in this table, which is too far from what we studied between our basically 1,000 samples. So if we do the basically interactive histogram, you can see, for example, in our first try, for example, this is the TVD that we get if we take the one samples. Then if we go up, this is like another one. you can see distribution as getting more basically more real, that as the more that we try. Okay, this is like 51 try. So if we go up, you can see now it doesn't change much, but it's getting closer to what we observed earlier. I mean, considering all the 1,000 experiments here. So you can basically vary this and see how it changed, but it doesn't get close anywhere around basically 0.14. So this was that's the basically original one. So questions for you now is how do you think it changed? Okay, how do you think our analysis would change if the jury panel had only 50 people? You remember here we picked 1,453. Okay. So what if our jury were only 50 people? Can you think about it and say how you think it would be basically our result would vary?
I think the TVDs would be a lot more spread out in the graphics, because since the sample we have is a lot smaller, only 50 people, the chances of having a correct representation of the population within the sample is much lower.
Okay, that's a very good guess. So here let's run it and see what we're going to observe from running it. So I'm defining this table. I'm doing the same thing. Now I'm just selecting 50 people. So this is the distribution I get from running it one time. So these are my basically the real data. These are the real data. These are my society or my county people, eligible people. And these are the random samples that I got. Okay, if I run it like another time, you can see it varies more. Okay, but it's still not that much. Okay, but it varies much more than what we saw earlier. So if we do the same thing, we run the experiment for 1,000 times and we study that. And then let's just draw this. This was basically the experiment. Can you see everything?
Yes.
Okay, maybe a little bit better. So you can see this is the earlier experiment with 453 people in my basically jury panels. These are the one that we did with 50 panels. So you can see, as you guessed correctly, it's distributed much more, because, for example, if I run it like one time, it's here. If I go higher, you can see it varies a lot. You can see it's now distribution is getting basically the variation is a lot. So you can see how it varies. Okay, so now considering this, then and the TVDs of this TVD, if you look at this, then you see TVD falls here. So now it's not basically how can I say? It's not impossible to get that type of distribution, right? Because the TVD was like 0.1514. It's like around here. So you can see it's now would be possible. Okay, so is it any questions so far? Is it clear? Okay. But anyway, in this case, as I mentioned, let me show you again so you can recall what happened. This was the study happening in between 2009 and 2010. Okay. So that was the study period, but the data that they were using in that study was from the consensus that they did in 2000. Okay, in 2000 they did, and they got basically all the data. That was a distribution of the people in 2000 basically data, and they were studying that in 2009, 2010. But then in the basically census of 2010 that they did, they find this distribution in the society. You can check it. Now whites are basically less, 42%. African American are 12.6. Asians were 26%. Okay, is I think native Haitian, and they were like less than 1%. Other races, so you can see the distribution was different. Okay, these were the panels that they studied. Okay, so you can see this was the that time this was the eligibility, this was the panel. But now that the population basically changed. Okay, so what we can conclude is our all analysis is only as good as the data you start with. Okay, because the data was a little bit older, so I'm not concluding anything here that the jury panel was fair even still. Okay, but you can see the distribution change in that 10 years period. Okay, because you remember we were talking about the census, that each 10 years we do the census, we count the people, we basically gather the information in our country. So that was the basically data from 2000 that they were studying in that experiment. But in 2010 census, they got this distribution. So you can see Asians increased to 26%, and in the panel we had 26% Asian. So now I'm not going to basically analyze this further, but the data is there if you want to play around with it and do the same experiment again and see was it really fair or not. You can do that study. Okay, any questions?
No.
Okay, let's go back to our slide. So let's talk about hypothesis testing. So basically what we were doing so far was sort of hypothesis testing. We had this hypothesis that, for example, the jury panels are fair, okay? Then we did a study to make sure that was the case, okay? So that's basically we were doing the hypothesis testing. But in hypothesis testing, there are certain basically steps that we follow always. So let's talk about them. The first step is basically defining the hypothesis. It is a test chosen between two views of how data were generated. In that example, for example, we said the jury are selected randomly. Okay, that was the data. The data was our jury data, and we had two different basically view of how this data generated. One is like randomly, so everything is fair. Another one was the data is not basically, the jury is not selected randomly, totally randomly. So the null hypothesis is the process that data were generated at random. That's our null hypothesis. And our alternative hypothesis is that it proposes some effect other than chance was involved in selecting the juries. So then step 2 is to test the statistic. Basically, a value that can be computed from the data and samples for samples that are basically testing statistic, and then the sampling distribution of the test statistic. So we want to basically know what the test statistic might be if the null hypothesis were true. So we considering the null hypothesis is true, then we do the test statistic and we study if is it our data basically rejecting our null hypothesis or not. So then approximating the sampling distribution by empirical distribution, that's what we did exactly. We empirically studied our juries by trying that experiment like 1,000 times and running it and seeing what type of data we gain from there. So in this example, our null hypothesis was the panel of jurors was selected at random. Alternative was the panel was not selected at random. Then the test statistic was the TVDs between a panel and the population distribution. That was our test statistic that we studied. And the sampling distribution of the test statistic was what we did. It was simulate drawing panel from population, see if the actual panel TVDs is likely with what we see in the real examples. Any questions so far? Okay. So, okay, let's do some experiment, for example. So what I want you to do is, I appreciate if you turn on your camera, guys, because I need to see you to do this, okay? If you can do that. So I want you to pick a number between 1 and 20 and just remember the number. Any number between 1 and 20, okay? Just hold on into the number. I'm waiting for other people to show up. I'm looking here because this is where I see you, okay? So pick a number between 1 and 20, any number between this, okay, you got it? Okay, so let's say, for example, that number is your, don't change it, okay? That number is your weirdness level. Minimum is like 1. The lowest is like 1, you're less weird. If 20, closer to 20, you are the most weird. You are more weird. So for example, if I do the same, I did the same, I got 17, my weirdness level is high, okay? So I wanna know what we did. So what I'm going to study is if there is a chance that I have been selected from this class randomly, okay? Clear? So who picked the basically number minimum, at least 1? Can you hands up? At least 1. At least 1, so everyone should hands up. I mean, because that's the lowest one, right?
Yes.
So yeah, I'm going to count. So can you raise your hand so I can see? Ricardo Daniel, you didn't pick any number?
He's not paying attention for sure.
I picked a number.
Okay, who took at least 5? At least 10? I mean 10 and above? Like 15 and above? Like 16 and above? Like 17 and above? So anyone pick 17 and above? Okay, we have like me, Oscar, and Hector. So and Rafael. So we have 4 person picking 17 and above. Okay. So if you consider we are like 20, how many? 23 person? 4 out of 23. Anyone can do the math? The percentage, the chance that I've been selected randomly from this group. So you got what's happening? It's like I select basically a random number. Okay, we have like 17.4%. Someone's basically sending there. Diego saying 17.4. So basically, I pick a random number. Let's say my weirdness level was 17. So then we studied the class and see if there is any chance that I can be selected out from this class. So we see there is like 17% chance, or like 0.17, the chance that I have been selected randomly from this class, okay? So let's talk about that more. So here, basically, the hypothesis, the null hypothesis was Maddie's, like my weirdness is randomly chosen from the population of this data science course. Alternative hypothesis was it's not selected randomly from this class. So the test statistic was weirdness. The higher number was more weird. Okay, so that's what we did. Because this is my basically the whole, how to say, the whole society that I'm studying is this class. Okay, so I could just do the test statistic by looking at the classroom, right? Because I have everything. I just asked you to raise your hand, all of you to raise your hand. So that's what I did. Okay, I don't need it to go and pick randomly, so don't confuse that. So that was the null hypothesis that picked randomly from this class. Then alternative was no, that's not correct. So we studied that. Okay, so that's another example. So we're going to talk about that. You're going to see how that's related to what we're going to talk. I don't know how many of you are familiar with Gregor Mendel. How many of you heard his name?
Me.
Okay, one person. Oh, okay, two persons. I see. Yeah, Carol and Brian. Okay, basically, Gregor Mendel was an Austrian monk. So he had not much things to do. Okay, so he was studying the plants and like observing the plants and their colors and size and things. So he was basically a founder of the modern field of genetics. He had basically a huge impact on that. So among many experiments, he tested the hypothesis that pea plants will bear purple or white flower at random if we do this, okay? If you have like purple, we have white. If you do the self-pollination, you get purple, you get white, then here, okay? Then if you do the cross-pollination, you might get all purple. But if you keep doing that experiment like more like generations, at some point you're going to get like 3 out of 4 as pink and 1 out of 4 as a white, basically, flower. Is it clear? So that's what he studied and what he observed. So he basically observed that 705, you get 705 purples, okay, comparing to 222 white flowers, okay. So that's what he concluded, that the ratio would be 3 to 1, okay, between white and purple flowers. So basically, so we're going to study this and see how correct was that. So this is basically our null hypothesis, okay, that the ratio would be 3 to 1 between pink and white flower. And the basically alternative hypothesis would be Mendel model does not represent the real world, okay, in the case of pea plant. Is it clear so far? Okay, let's study that.
Teacher, I have a question.
Yes.
I didn't understand the part about the alternative.
Which one?
The
alternative
part.
Okay, we said we have, like, for example, we have 2 different views from our like, let's say like this example that I mentioned, like the weirdness level. Okay, very good. So basically we are having like, let's say we are choosing 2 view of how data were generated, okay? In a jury example, our null hypothesis was the jury are selected randomly. And the alternative hypothesis was, no, they are not selected randomly. Okay, so that's basically null hypothesis and alternative hypothesis. Is it clear? Or in another example that we studied, like this weirdness level, the null hypothesis was like, my weirdness level is selected randomly from our class. That's a null hypothesis. And the alternative hypothesis was, my weirdness level is not selected randomly from the class. So here in Mendel's case, we say the null hypothesis is this, we're going to observe this 3-to-1 ratio. That's basically representing our real world. What we're going to see, that's our null hypothesis. The null hypothesis is the Mendel's model is correct. Basically, the alternative hypothesis is Mendel models is not correct, or it doesn't represent the real world. Clear?
Okay, thanks.
Yes.
Okay, let's go see what we get here. But let's have like a 7 minutes break, okay?
So,
Don't get disconnected. You may turn off your camera, you may mute, and we come back at 11 a.m.
Okay.
Okay. Even if you basically turn off your computer, come back tomorrow, still the session is running. So you don't need basically to restart the kernel, go through that pain. It's much easier. Okay, basically the session is always running for you. So you can just come, continue coding. It's much, much easier if you do it locally. Any questions? Doubt, recommendation? Okay, let's continue. Where were we? Okay, so we were talking about hypothesis testing and the Mendel, Igor Mendel model about his observation. So if we basically this is our null hypothesis, okay? This is our null hypothesis. We have basically 3 out of 4 purple flowers and 1 out of 4 white flowers. So we put this table basically in this array in our null hypothesis. So we can, for example, we can select randomly 10 from this this array. So this is basically what I get. If I count them, for example, count the purple one, you can see I get like 8 purples here. Okay. So if I run it again, I get 7. If I run it again, I get 9. So in order to see how basically this 75 was basically the Mendel's observation. 75% of the time you see purple, and like 25% of the time you see white. So if I get these proportions of purple and basically minus what Mendel observed, basically I can get how varies is my data from his data. Okay, right? Is it clear? So,
Yes,
pardon?
yes, it's clear.
Okay, so if we do that, basically I put everything here. So if I can run it, this one. So what I want to do here is like if I remove this and run this a few times. So the closer I am to the 0 means basically my observation is also same with Mendel observation, right? Because I have to get proportion of purple, my proportion of table should be also 0.75, right? Because if I take randomly, then it means I'm observing the same thing that he observed. So if I run this few times, we can see it change. It goes to minus because now my proportion of table is smaller than 75%. Okay. If I run now, my proportion of purple is bigger than 75%. So what I want to do is to get this absolute value here. Okay. That doesn't matter for me. I just want to see how different is my observation from Mendel's. So let's do the same thing, defining basically the same thing in a functions. These are the total plants that he counted. Okay, as I show you in the slides. This is what he observed, okay? He observed like 705 out of 929 were purple. So this is what he observed. I put it there. And the observed statistic basically was what he observed minus that what he concluded, that 3 out of 4 are purple, okay? So if I basically get this value, this is what he got, okay, in observed statistic. Like this 0.0088 was his observed statistic. So this is what I get from running this my experiment here. This is what I'm observing. Okay, so let's do that not just one time but multiple times. So what I'm doing here, I'm doing the same thing. I'm picking a 10 and I'm basically running it 500 times, 5,000 times, and I'm calculating my basically this value. Okay, this proportion minus basically is 0.75 that he observed. So in order to see how varies is my basically data. So this is the null distribution that I get. This is basically I call it null distribution. So what I did, I get this one, okay, and I minus that value. I use this basically this function, okay. So these are what I'm observing. If I put that again in the histogram to see it better, this is what I've observed, okay. These are the distribution I'm observing, okay. So you can this line, blue line, is what Mendel observed. This is his value, okay. This is that value. Where was that? This is this value, okay. The observed statistic. This is the observed statistic, okay. So you can see these are the result of 5,000 experiments, and this is what Mendel observed. So just by looking at the graph, you can see that basically what you can conclude from the graph, from this histogram. So I mean, we talk about null hypothesis that Mendel's basically observation describes the board. The alternative hypothesis was no, it doesn't describe the board. So what we conclude from here? Does it describe the board? I mean, are we proving the null hypothesis or are we rejecting it? Any comments?
Rejecting?
You're rejecting the null hypothesis?
I think so.
So why is that? Can you tell the reason why we're rejecting null hypothesis? So you can see these are the basically mostly are falling here, okay? So, and his observation was basically somehow accurate. If I
think the observation is like the mean of the distribution, maybe.
Pardon?
I mean, if we get the mean, maybe the result would be like a value very close to the observation.
Okay, true. So by saying that, are we proving the like null hypothesis?
I think we are proving it. Yes.
Okay. So let's talk about that further a little bit. Let's go back to the slides. So let's talk about p-value. I'm almost pretty sure you heard about it. Maybe you know already what is it and how it stands. Or probability value or p-value. Basically, you can define it like this: the p-value is the chance under the null hypothesis that the test statistic is equal to the value that was observed in the data, or is even further in the direction of the alternative. Are you confused enough with the description? Anyone of you knows p-value before? I mean, you studied? Okay, I think majority you studied this. Okay, let's look at this. It's maybe when you read this, it's kind of like not easy to understand, but it is really, really straightforward concept. Basically, can you, okay. Basically, if I get the sample state that I did, okay, and then I'm comparing it with the observed statistic. These are my observed statistic. And I'm comparing if my sample stat is bigger or equal to my observed stat, then I'm dividing it basically by the length of my sample stat, and I'm getting this number. What this number says is like 54% of the results are higher than my observed stat. Basically, 54% of the data are falling in this part. Okay. So that's basically its p-value. I'm calculating here the p-value. The p-value in this example, like considering the observation that Mendel had, is like 54% or like 0.54. If, for example, let's say I tried the experiment, okay, I did it again, and now I'm doing the same thing. Now this is, if I tried another basically experiment and I get this observation, and I draw the histogram with my experiment, and I put the line here, you can see this is what I get, okay? This is the line from my observation, imaginary observation, okay? And if you calculate the p-value for this, it would be 0.03. Okay. Any questions so far? So this is what I did here is like, I just imagine I started, I don't know, I planted some pea beans in my backyard, then I started counting the flowers, and I did the experiment, and I observed this is the ratio between basically the pink flowers and white flowers. Okay, so this is what I observed, imaginary observation. See, if I do the same thing experiment, I put the my observation and show it with the histogram, and I calculate the p-value. You see, this is the p-value for my observation. So you can see the chance of, basically what it says, the chance of occurring Mendel observation is 54%, and the chance of the occurrence of my observation is 3%. Okay. So another question here. So which one is more significant? If those that they know already what's p-value, which one is like statistically significant? Any comment?
Can you repeat, please, the question?
I mean, what of this observation is statistically significant? If you don't know the answer, it's fine. No worries.
Well, the both are significant because it depends on what you're looking for. Mm-hmm. Because in the first you for like the half of the distribution, and in this observation you look below the 0.03%.
Mm-hmm. Basically here it means the chance of my observation to happen is 3%. The chance of observation of men that happened is 54%.
I
Okay.
think that the first is more significant because the p-value is bigger, maybe.
Okay. So, okay, let's talk about it. The p-value is a little bit like confusing when we're talking about that. Let's go back to the slide and see what we have. So, talking about—
Professor, I have a quick question.
Yes?
The p-value represents the quick definition would be the probability of our null hypothesis happening, right?
I didn't get the probability of what?
Of our null hypothesis happening.
Yes, true.
Okay.
Yes. Yes. Yeah, you can define it like that. You can say that. So basically, okay, let me correct myself. It's not probability of our null hypothesis happens. Is considering our null hypothesis is correct, that's the probability of our observation. That the earlier answer was not precise. That's not correct. We cannot say that. So be considering if our null observation is correct, what is the probability of that observation? So we have this, let's talk about this convention of consistency. I mean, this number I'm showing you here is not really based on any mathematical proof or observation. It's just, I'm going to talk about how we come up with this 5% and 1%. Basically, if let's say there are, like, we have two conventions talking about in the tail. If the area in the tail is less than 5%, the result is, we say the result is statistically significant. Okay. And if basically the area in the tail is less than 1%, we say the result is highly statistically significant. Okay, let me go back to this and see what we're talking about. So basically, looking at this, okay, we are looking at the tail of our distribution. So that in the tail of our distribution, this is the value, is 54%. Basically, this 54% is, if you add all this area together, because you remember we were talking about percent per units, you get this percent per unit level, you multiply this area here. If you do that for all of them, you get this value. That's our p-value. So we're looking at the tail, okay? And here, if you look at this is the tail. This is like 3.5%. So here, what we're saying, if we're saying, basically talking about, I mean, don't confuse this, okay? When we're talking about the what is statistically significant, is we're looking at the tail of our distribution, okay? And if it's below 5%, this is just an agreement between people. I'm going to say where this 5% is coming from, or why this 1%. So if it's less than 5%, we say the result is statistically significant. And if it's less than 1%, we say highly statistically significant. What it means it's really less likely I get that result, right? If we consider this example, it is really less likely I get this result. That's why it is a statistically significant result. So this, I mean, this p-value, I mean, if you didn't get it totally in this class, please, I insist, keep searching about it, look at other videos. It's very important topic, because I don't know what you want to do, but you always, statisticians talk about p-value. That's how we decide about the result. That's how we discuss about the result. That's how we discuss how accurate are our results, how significant is the result that we obtain in our research. So even if you see studies in newspaper, they usually refer to this value. That's how basically you can say a study is significant or not. A result is statistically significant or not. So what it means is here, it's very less likely I get this result. So it is significant. Here, I'm just talking about the significance, okay? Here, it is not that significant. I mean, talking about these numbers, because it's statistically almost definite I get this result, okay? I have 54% of getting this result. It's like even higher than a coin toss. So I definitely get this like very much high chance of getting this result. So it's not statistically significant, but getting this result, for example, talking about the jury, maybe I have to make it more clear, talking about the jury example, like it was very less likely if we, consider, okay, let me clear this. We are talking about that our null hypothesis is correct. In the jury example, our null hypothesis is the jury are selected randomly. So we say, okay, this is correct. So now let's study that. Then we studied, then we got a result that is very, very less likely to happen, right? If we say if you pick the jury randomly, it is very, very less likely you have 8% only black. So that observation is statistically significant. So it means something happened there, okay? Either we reject the null hypothesis, or that is basically, we can reject the null hypothesis. We can say, no, the jury is not selected randomly. So that's why it's statistically important, because we can, based on that, we can derive some conclusion from it. I hope it's clear. It's maybe a little bit confusing, but I hope it's clear. Any questions? So that 5% and 1% are coming from this guy. Sir Ronald Fisher was not that old. Basically, you can see it's like he was early 19, basically scientist. So he said we have the duty of formulating, of summarizing, and of communicating our conclusion in, let me say, in intelligent form, in recognition of the right of the other free mind to utilize them in making their own decisions. Don't try to remember it. It's just, he was saying, like, present your data and let other people to decide if it's basically what drive the conclusion from it. Then he says it is convenient to take this point, like 5%, as a limit in judging whether a deviation to be considered significant or not. And then it says if 1 in 20 does not seem high enough, odd, we may, if we prefer it, draw the line at 1 in 50, or like 2% point, or okay, or 1 in 100, or 1% point. So that 5%, 1% are not really based on any statistical or based on any mathematical calculation that they come up with this magical number of 5% to define if a study is significant or not. It's just his suggestion that got popular among the statisticians and among all the scientists. Basically, we are sticking with this. But this 5% and 1% are not always fixed. It might differ in some area of, like, for example, talking about medical science, they might go with this 1% because they want to be more, basically, they want the result to be more accurate, more reliable. In engineering, usually in our result, that is our type of study, we stick with that 5%, okay? And like in some other science, maybe like social science studies, they go even with higher percents for p-value to consider a study is significant or not, data or an observation is significant or not. They go 10%, 15%, but you can consider that 5% as a significant result. So this is the same thing, that observation we saw. This is the observed statistic. This is under the null hypothesis. If you consider the null hypothesis is correct, This is the probability of it. And this area is the p-value. You remember we show you 5%. If you draw a line here, you get this 5% here. That's you consider that your p-value here. So you can define, you can basically discuss about the p-value in a slightly different manner, talking about error probability. So can the conclusion be wrong? Of course, the conclusion sometimes can be wrong. So it can go wrong like two ways. Okay. For example, we test our test reject the nulls. Okay, while null is true. So that's basically a wrong conclusion. We did some tests and we rejected the null hypothesis, but the null hypothesis was correct. Or we test our test doesn't reject the null while the null is basically while the alternative is true. So basically, this conclusion can go wrong in these two ways. Okay, either your test rejecting the null while null is true, or your test doesn't reject the null while alternative is true. Okay. So basically, the p-value basically can happen here. Okay, when our test rejecting the null, but the null is correct. Maybe in that in the second example that we saw, let me go back. You remember this example? Okay, in Alameda County in California, we studied. We thought we are doing something wrong here. Then we get the latest census data, and we see, oh, now the population is look like same. For example, Asian are 26%, and for example, in our panel also we had 26%. So what we did here, based on the data we had, this mosquito killing me. Based on the data we had, we observed, we thought maybe the null hypothesis is not true. Okay. Then we check the latest data here. We see, oh, considering now this data that is up to date, the null hypothesis look like true. So that type of problem can happen. So p-value would basically can, let's say, help here or not. So basically the cutoff for the p-value is an error probability. For example, if your cutoff is 5% and the null hypothesis happen to be true, okay, you don't know that, of course. So there is about 5% chance that your test will reject the null hypothesis anyway. Is it clear? Okay, perfect. So that's all of it from my talk. So let me see what I have here. Okay, there is this discussion question. I don't want to spend a lot of time here. So what happened if you remember we consider that absolute value? Okay. What if I remove that absolute value? You remember here, I consider this absolute value. Okay. What if I don't consider the absolute value? How my data going to look like? Any idea?
It's gonna be like a normal distribution.
So you mean it look like a normal distribution?
Yeah, like a Gaussian. I don't know if that's how it's said.
Any more idea? Any more observation? If I run the same thing, as you guess correctly, we get basically like a normal distribution. Okay, so I don't want to, I just included here so you see how it works. Basically, we got the absolute value. So all this, basically, these data are falling the other side, right? All this, like, this data would fall here. That's why we see this type of, basically, observations. If we don't, we get this, still we can conclude, or still we can drive the same conclusion from the same data, okay? Doesn't change anything, basically. So let's, okay, we have this lab 6 activities, okay? We have the lab 6 activities. It's already online in the, where is it? In the server. So you can check it out. But before we go to this, I want to discuss the COVID-19 experiment that you did, the homework of COVID-19. So let me see if I get, these are the basically submission I received, okay? So can you guys, I want you to, all the submissions, I want you to share your screen, okay? One of the team members, no matter either of you, okay? Share your screen and talk about what you did in that basically experiment, like very short, like 5 minutes, okay? Okay.
Okay.
So let's start with the first one. You can see, I don't know the name, I'll just put the codes here. So can you please, this group? What is this group?
I am, and also Miguel.
Okay, so one of you, can you share your screen to talk about it and like run it as we are going through?
It doesn't run in. Can you share it, Miguel? Because it doesn't run really.
I don't have occupying this computer. I have in the other one, so I can share my screen.
You cannot.
No,
Okay.
it's in the other computer.
So Diego, it doesn't run for you?
I have problems with the table because I upload to another table, more recent table.
Okay.
But it doesn't run. Also, your example, it doesn't run well with the link to the table. I have problems with that, but besides that, it's I can run it, I think so.
Okay, just hold on. I'm going to stop recording here. So I want to basically split the videos in 2 parts. The first part, that what I discussed now. The second part will be talking about labs and these activities. I record that too. I upload it separately so you can check them separately.